Hi everyone. This week we talked about the genetic diversity in the immunoglobulin and MHC complexes. So first for the MHC, there are two different classes, 1 and 2, and each of them have separate genes. Class 1 has 3 genes, HLA-A, HLA-B, and HLA-C. There are some lower ones too, but their function is not really well understood, so we won't talk about those. Class 2 has 3 pairs of genes, so 6 genes total. HLA-DP, and then it's A and B, HLA-DQ, A and B, and HLA-DR, A and B. The MHC genes are passed down as a single haplotype, meaning that they are grouped in a set of so both the class 1 and class 2 molecules. They are expressed codominantly, meaning that each individual receives one from their mom and one from their dad. So this creates diversity because there's no possibility that you would receive both haplotypes or both molecules from your dad or both from your mom. And this also indicates that a single person can express up to 6 different HLA class molecules, and so that's interesting as well. The MHC molecule is regulated coordinately, which means that all of the molecules are expressed at the same time on a single cell, and any factor that has an effect on the molecules will affect them all. So it's not like just one gene will be affected, they all will. The polymorphism is a huge presence in the MHC. Genetic variation, this means that there is the presence of genetic variation, and it's very rare that someone will have the same haplotype as you. It's almost guaranteed that the haplotype from your dad will be different from your mom, and so there's no very rare chance that you would end up with the same one as either of them. This also means that within siblings that there's a rare chance of being identical. I'm one of 5 children, and there's probably 0 chance that we all have, or that we have the same haplotypes, so I think that's really interesting. And so that creates a lot of diversity within the MHC genes. So then for immunoglobulins, they have a crazy amount of diversity. Each individual has the ability to generate 10 to the 11th or more molecules of immunoglobulin, which can then be rearranged and recombined to produce millions of possible sequences. It's hard to even comprehend how many different possibilities of immunoglobulins we can have. So I'm going to quickly describe the synthesis of the light chain and the similar processes for the heavy chain. So early in B-cell development, one variable gene combines with one of the joining segments, and this mechanism is at random. Then there's the reg 1 and 2 complexes that cut the immunoglobulin DNA, and so without these molecules, a person would lack mature B and T cells, which would create a lot of immunodeficiency problems. So this means that anything between the variable and joining regions is cut out, and then the DNA is rearranged, the primary RNA transcript is made, and so then at this stage, the variable, joining, and constant regions are all brought together and are ready to pair with the heavy chain. So all of that can create a ton of different rearrangement in the orders because there's different variable genes, different joining genes, and then the constant chain is pretty much the same. But so that alone can create a lot of genetic diversity. The heavy chain can create more diversity because it has an additional diversity chain, so that alone can make even more possibilities. And then combining the heavy chain components with the light chain, it just creates millions of opportunities for changes. There are a bunch of other ways that they can create genetic diversity as well. There's the combinatorial association. So there are 40 variable and 5 joining genes for the light variable region, which can result in 200 different chains for the light, and then for the heavy chain, it's got more than that, to about 6,000 different heavy chains. And then there's also junctional diversity. So at each site there can be improper joining, and so that can, like with different nucleotides, so that can cause different pairs, and class switching that creates different immunoglobulins. And then importantly, they wanted to talk about affinity maturation and somatic hypermutation. So somatic hypermutation is a random point mutations in the recombined VDJ unit in either the heavy or light chain variable regions, and this occurs at a much higher rate than any other mutation. So that's why it's hyper meaning more. And the affinity maturation comes about because of somatic hypermutations, and this can result in the increased affinity of an antibody to an antigen. So generally low affinity antibodies are produced, but with the somatic hypermutations, the mutation to increase affinity occurs quickly. So that was kind of a lot of information, and I definitely learned a lot about these different MHC and immunoglobulins genetics.